Hi everyone, in this video I will be going through the TE for gene expression and mutations. Let's begin with question 1. Question 1 asks you which option is correct, and they give you an image labeled P, Q, and R. Alright, so how do we do this question? First and foremost, when we look at P, this refers to an entire chromosome. There are two kinds of chromosomes, right? We have chromosomes that look like just one single structure, and after S phase or after DNA replication, it will become two sister chromatids attached to the centromere. So each one of this is called a sister chromatid. However, when we look at the entire structure P, that is a chromosome. This is also considered a chromosome, right? So the difference between a chromosome before S phase and after S phase is that after S phase we have two sister chromatids attached to each other. Alright, so that means only option A or option B can be the answer. Usually when we do MCQ questions like that, you can immediately rule out a few options already. Let's go to Q. Q is referring to this entire structure. Alright, and so an entire structure will have to be DNA. How do we know that it's DNA? Because we see it's a double helix. Finally, when we look at R, R is just referring to a sequence of DNA. Alright, and a gene is a sequence of DNA. Okay, so the answer will have to be A. Next, let's go to question 2. Question 2 says, which statement describes a gene? Let's look through all the options. A base with a sugar and a phosphate group. Alright, what is this referring to? A base, okay, so maybe a base, it could be adenine, attached to a sugar, okay, and a phosphate group. So A is referring to a nucleotide. I say again, A is referring to a nucleotide. Okay, so therefore it cannot be A. Which statement describes a gene? A number of DNA molecules. That's not true. So a gene is not a number of DNA molecules, right? It is referring to a sequence of DNA. So in other words, you can also say that a gene is a sequence of nucleotides. A sequence of nucleotides means, if we look at A, T, G, C, 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 and so on and so forth, a gene is simply a sequence of these nucleotides together. Could it be the chain of alleles on a chromosome? So let's say we have an entire chromosome here, and they're saying a chain of alleles, right? We can have different alleles on a chromosome. This allele could be allele A, this could be allele G, this could be allele maybe M, and so on and so forth. A gene does not refer to a chain of alleles. It just refers to a sequence of nucleotides or DNA. So the answer is C. Next, let's go to question 3. Which option correctly describes the effect of nonsense mutation? Okay, A, no effect on resulting amino acid. If there's no effect, we refer to it as a silent mutation. If it increases the length of the amino acid sequence, I would say very likely there was some sort of an insertion, okay, that resulted in the increase in length of amino acid. When I say insertion, I mean insertion of base pairs. Okay, that means, let's say we have, I'm just going to write out a sequence, maybe AAA, GGG, CCC, right? Each one of these refer to a codon. So in other words, each codon can translate to an amino acid. So maybe this is amino acid 1, amino acid 2, amino acid 3. If I have an insertion somewhere, right, let's say I insert TGC, that means to say now I've inserted also one additional amino acid, and then my amino acid chain will be longer. Next, let's look at C. Replaces one amino acid for another amino acid. This is referring to a missense mutation. Missense mutation is a swap of amino acid if we change the codon. Last but not least, D. It replaces one amino acid for a stop codon. Okay, and that is what a nonsense mutation is. It refers to a change in a codon become a stop codon. And I think one of the stop codons is UAG. Okay, so the answer is D. Let's go to the structured questions. The first structured question uses a case study of cystic fibrosis, which is a genetic disorder. It says that this genetic disorder has a deletion of 3 consecutive nucleotides. In other words, if we say it's 3 consecutive nucleotides, we're saying that an entire codon has been deleted. As a result, the patients will suffer from excessive production of thick mucus and so on and so forth. So the question asks you, explain how the 3 nucleotide deletion results in a non-functional protein. Okay, first and foremost, as I was marking many of our scripts, I realized that many of us think that a 3 nucleotide deletion can result in frameshift mutation. Okay, I just want to show you why it doesn't really result in a frameshift mutation. Okay, let's say I have a gene that consists of maybe, let me just write this down, maybe it's ATC, maybe GGG, CGC. Okay, so let's just work with 3 codons. Here we have codon 1, codon 2, codon 3, right? This will result in amino acid 1. Okay, maybe let me not use amino acid 1. I shall draw out some cartoons. Okay, let's say the first amino acid is a triangle, second one is a square, and the third one is a circle. Okay, and if we were to translate this, we're going to get a polypeptide with these 3 amino acids. So what happens if we delete 3 nucleotides? Okay, let's say we delete 3 nucleotides and we delete GGG. This would mean that my resultant polypeptide is going to just have a diamond and a circle, right? Where did it come from? It came from this. But you notice the amino acid did not change, right? Yes, the second amino acid now is a circle, not a square. By the end of the day, there was no frameshift mutation. Okay, so what does it mean by a frameshift mutation? A frameshift mutation means, right, that our reading frame has changed. Okay, what do I mean by that? Okay, we look at the same sequence of DNA here. Okay, and first codon, second codon, and third codon. Let's say rather than deleting 3 nucleotides, I delete just just 1 nucleotide. Okay, this becomes a little bit problematic because what used to be the reading frame is used to be like that, right? ATC, GGG, and CGC. Now I'm going to read it as GGC. Right, what happened is that now I'm shifting the reading frame. Okay, then over here now it will be GC. Okay, and perhaps maybe what was downstream, right, is maybe an A, so now A. Right, so what has happened is that the reading frame has shifted. Okay, because we are now missing a G. The reading frame has shifted. So originally, right, we would have gotten a diamond amino acid. Originally, we would have gotten a square amino acid, but now, because it's read as G, G, and C, we may end up getting a different amino acid. Originally, we read this as CGC, but now, because of the frameshift mutation, now we read it as GCA. And this may result in yet another amino acid. Maybe now it's a triangle amino acid. As a result, the polypeptide I formed is vastly different from what was the original. The original was a diamond square circle. Now it's a diamond heart triangle. Right? So when we refer to frameshift mutation, right, there is usually a change in the reading frame. And the codons will be read very differently. However, notice if we were to delete in multiples of 3, we don't really change the reading frame. The reading frame still is the same. We can still read the same amino acid after the one that's deleted, everything still remains the same. Yeah. In other words, if we delete in multiples of 3, it's just like if we have a sentence, we just delete one word, and we can still read the sentence just without the word. But when we remove in multiples of 1 or 2, in this scenario over here, we end up getting a sentence that don't make sense. Yeah. So a frame shift only happens when we delete or insert in multiples of 1 or 2. But in multiples of 3, everything downstream of what you delete or insert will still be read the same. You still get the same amino acids. Okay, so in other words, we won't be using the word frame shift. And also that also means that you won't get any missense mutation or nonsense mutations, because nothing downstream will change. Okay, nothing after this deletion really changes. Everything is still read the same. Originally circle, still circle. Right? So you won't expect any missense or nonsense. Okay, so let's look at the answer. So what happens when 3 nucleotide deletion happens? Since, the DNA has been mutated, what will result is that the transcribed RNA will have a loss of 3 nucleotides. Okay? If you delete in your DNA, what will happen is that your mRNA will also be missing 3 nucleotides now. This means that you will lose one entire codon on the mRNA. If there's an entire codon missing, you will also be missing now one amino acid. Okay, and what will this result in? Well, this will result in a change in the primary structure. If there's a change in the primary structure, very likely the kinds of bonds and interactions that will form will change, and this will affect the secondary, tertiary, and quaternary structures. And if these levels of structure all change, you also change the 3D shape of the protein. And since structure determines function, a change in the 3D structure may result in the protein becoming non-functional. So to summarize, if your DNA changes, your mRNA will change. And if your mRNA changes, you'll result in a different primary structure. If the primary structure changes, it can affect your secondary, tertiary, quaternary structures, and this will affect the 3D shape of the protein as well as the function, because the shape determines function. Okay, so you can pause here if you want to have a look at the answers. Next, let's look at question 2. Question 2 gives you an example of sickle cell anemia caused by the hemoglobin variant. Right? And also they show you 2 tables. They show you a table of beta globin sequence in a normal adult and beta globin sequence in a mutant adult. Right? We notice that largely all the amino acids are the same except for amino acid 6. Okay, so that is the information provided. Let's look at the question. They ask you to state and explain the type of mutation responsible for this change in amino acid sequence. Okay, if we look at the top over here, we notice that everything after amino acid 6, 7, 8, 9, 7, 8, 9, actually they are exactly the same. Alright? If things after amino acid 6 remain the same, it means that it cannot have been a frameshift mutation. Okay, this is out of the question, because usually frameshift will change a lot of things. There is also no nonsense mutation. Okay, and therefore, since this change is so localized, it's only affect amino acid 6, it means very likely the change was only unique to this particular codon that coded for amino acid 6. That means to say, likely some form of substitution occurred or missense mutation. Missense mutation means the amino acid changed from one to another. And usually this happens when we have a substitution. So many of you did quite well for this. You said that it is likely a substitution or a missense mutation. Okay, we usually don't say it's a substitution mutation. Usually we say nucleotide pair substitution, that's like the process. But we describe the mutation as a missense mutation. I ask most of you to just elaborate what exactly you are substituting. If you do a little bit of research, actually the base that's being substituted is specifically from thymine to adenine in the DNA. And if you substitute the base, you result in, after transcription, one of the codons to change from GAG to GUG. And as a result, the amino acid that's translated changes from glutamic acid to valine. And if you change the amino acid, you also end up changing what we call the primary structure of the polypeptide. Okay, so words that you want to use are substitution or missense. You want to elaborate what exactly was substituted in the DNA. Okay, what was substituted was thymine replaced by adenine. Okay, then you also want to mention what the amino acid change was from glutamic acid to valine. Then downstream you want to explain, how does changing the amino acid sequence affect the structure? Specifically affects the primary structure of the protein. Okay, and that's question A. Next, let's look at B. B asks you to suggest how this mutation in the hemoglobin may have resulted in fatigue and swelling. So there are actually two parts to this question. They ask you to explain the fatigue and the swelling. Okay, so to explain fatigue, you can say that because of the mutant form of the hemoglobin, it is inefficient in carrying oxygen, or maybe not so good at binding to oxygen. Okay, and hence there will be insufficient oxygen to supply to different parts of the body. Okay, that explains fatigue. If you want to explain the swelling, the sickle cell red blood cells have a higher tendency to block blood capillaries, or you can say obstruct blood capillaries. Okay, and if you block blood capillaries or obstruct the blood vessels, it can result in swelling because all the liquid accumulates in these parts of the body. Okay, some of you mentioned that that the red blood cell, sickle red blood cell, can adhere to each other and end up clumping or clogging the blood vessels. That's fine also. Okay, so two parts to this particular question. Let's go to this very last part. The last part, they show you the genetic code for 7 amino acids shown in the normal hemoglobin. So they are showing you what each codon would translate into. With reference to table 2.1 and 2.3, state the genetic sequence of the DNA template strand of hemoglobin A. Okay, so let's look at what we have so far. In your table on top 2.1 and 2.3, okay, what we provided you was only the amino acid sequence, right? Or in other words, you only provided the primary structure. Let me take this primary structure for this question. So we begin with leucine, I forgot what this is called, and proline. Okay, let's take these amino acids, these 1, 2, 3, 4, 5, 6, 7 amino acids. Okay, we're going to work with that. Okay, I have the same amino acids here. Okay, 1, 2, 3, 4, 5, 6, 7. Okay, so now we know the primary sequence. Let's work backwards. If we know the primary sequence, what will the mRNA sequence be? Okay, the mRNA sequence would be CUG, ACU, CCU, GAG, and so on and so forth. How did I get that? Well, I refer to this genetic code over here. Okay, so leucine will be CUG, and so on and so forth. Okay, so first thing I've gotten is the mRNA sequence. When you write out the sequence this way, it's actually from 5' to 3', because we read all mRNAs from 5 to 3' direction. Okay, so that means everything you see here is actually in 5' to 3' direction. Right? Okay, so next, we know that the mRNA strand is very similar to the non-template strand, right? Or another way to look at it is that the mRNA will be complementary to the template strand, right? because we use the template strand to synthesize, as a reference to synthesize the mRNA. So that means it has to be complementary. So if the first amino acid, the codon goes CUG, the complementary of it will be GAC. For the next amino acid, since it's ACU, the complementary will be TGA, and so on and so forth. The question asks you to write out the DNA template strand. So actually, if you write this out, you'll get the correct answer. However, many students did not write which is the 3' end, which is the 5' end. So you need to write 3' and then 5' at the other end. Okay? Then you'll get the correct answer. Another way to write this same answer is this way. Usually we will write sequences in 5 to 3 direction rather than 3 to 5. So I would write the template strand as AGA, AGA, then CTT, CTT, CTC, CTC, okay, another CTC, and then after that AGG, AGG, so on and so forth. Okay, so there are two ways you can write the answer. You can write it in the 3' to 5' direction, or the 5' to 3' direction. But don't forget to write the directionality so that we know which way to read it. Okay, so that is how you do this particular question. And if you go down all these success criteria, could you do a quick self-evaluation to see whether you understand how to do the various parts of this particular chapter. And that is all for this particular chapter. Thank you.